Well, it gives me absolutely great pleasure to be here at the start of another Bevan series talk. My name is Trevor Brandt, and I'm an assistant professor here in this department. And today's speaker, Rudy, has flown all the way out from Australia. And to give a formal introduction, I'm going to hand over to Professor John Horn to introduce him.
Thanks, Trevor. And for those who I haven't had the pleasure yet of met, my name is John Horn, and I'm a professor here at the School of Aquatic Fishery Sciences, and I also spend time over at the Alaska Fishery Science Center. We are in for a treat, I hope, from but just to give you a bit of pertinent background for Rudy's path, and not to end up directly here, but Rudy is a native of Australia, got his Bachelor of Engineering at the University of Tasmania, and then a PhD in Applied Physics at Curtin University of Technology. I'll give you just a synopsis of research interests, and I'm not editing any of this, reading it directly. Investigating reflected and ambient acoustic signals in the ocean with associated optical and physical sampling to make ecological inferences about the water column and the seabed, and more broadly to learn about ecosystem function and dynamics. Applying this understanding of ecosystem function and dynamics to the sustainable management of marine living resources. And as part of his research interest, he's been going to sea since about 1987, being both a participant and leader of our, what we say, surveys, what they call voyages, both offshore research, fishing vessels, and looking largely at Deepwater ecosystems and for assessment surveys. I have the pleasure of meeting and interacting with Rudy through the ICES FAST Working Group, which is a applied science and technology group affiliated internationally, not just European-based, looking at the application of acoustics and associated technologies to ecosystem management and assessment. And I think you're going to see a combination of the research, management, and technology through this. I hope you enjoy the talk.
Thank you very much, John, for that introduction. And Trevor and Sharif for inviting me here for the Bevan series. I'm very privileged to be here and to talk about something that I've been working in for the last sort of 25 years at CSIRO. I'm now a sort of a team leader of this Deepwater ecosystems, even though I come from a very engineering background. It sort of gives you an idea of how far my career has moved from the engineering that I started with years ago. And also I'll be presenting things from the rest of my team that's been looking at various areas of the Deepwater ecosystem. So I've divided this talk up today into three themes. One is the Deepwater habitats that we've been involved with around our fisheries. The second theme is the fisheries themselves, and in particular orange roughy. And lastly, I'll talk about something that we've been engaged, that we've developed recently, which is ocean basin scale monitoring of the pelagic worm. Given that the deep ocean probably represents 90% of the living space on the planet, and our knowledge of it is quite sparse. So I start off with just looking at the scale of the problem. You know, we're still trying to find out what habitats are really out there. And this slide I put together back in 2000 when we were starting to get into developing seabed mapping studies of our EEZ, and sort of the work was starting to come forward with marine protected area designs. And really we said, well, we don't know what's out there. And they were coming up with a lot of marine protected area designs, and we couldn't even tell them where the canyons were, where the seamounts were, or where the ecological hotspots were. And this is still evolving today, even though we've spent 10 years mapping some of our coastline. We still haven't found out, or we still don't understand all the habitats that we have. And also then we're looking at biodiversity, and a lot of our work over the last 10 years has turned into biodiversity assessments, looking at biogeographic provinces. And again, our knowledge of biodiversity and life in the ocean, deep ocean, is rapidly expanding and is definitely not complete. So you can see this is part of a continuum, and it's really a progression of knowledge that's going to take forth for many years to come. And I'm always impressed with this, that before the expedition in 1976, there was often thought that there was very little life in the deep ocean at all. So how much do we know about the deep ocean or the seascape? Our knowledge is evolving very quickly. Of course, the old adage that we know more about the topography of the Moon and Venus is still true today. But that poor understanding is rapidly increasing with the advances of technology. You can see here some of the maps that we had back in 1995. Then the first sort of SWOT mappers came in sort of post-1995, so we started to evolve our understanding. And then post-2006, we started with some very high resolution maps of the sea floor, and in particular the seamounts and a lot of the sort of the features where a lot of the via are attached. So at that stage, is around 2000, was sort of like a quantum leap in our area of gearing up for understanding the deep ocean. We developed a lot of acoustic equipment. We purchased a lot of swath mapping equipment on board the vessel. We developed a whole lot of optical sensors, and I'll start to talk about some of those results that culminates in probably 10 years of sort of applications of that technology. And predominantly, because this is has a sort of an acoustic sort of slant to it, that one of the big advances has been the move from single beam technology to multi-beam. And you can see, I won't talk about the other two, which are tow technologies, predominantly. This is operating from a ship. A lot of our earlier work was single beam, and we moved in the early 2000s to soft mapping sonars, and that has been the mainstay of a lot of our work. And that has multiple beams, comes across as a fan, you steam across, and it's a bit like mowing the lawn. You swath out and you get very high resolution imagery of the seafloor. But also you get reflectance from the backscatter, which tells you something about the substrate, and from that you can infer biological communities and where they might be. Another big advance has been with the optical sensors. We've developed a lot of high quality digital video footage. Some of that's used for science, but other parts of that have been used for communication with what's actually in the deep ocean. We've moved to very high optical cables and high resolution digital still imagery. And the digital still imagery is what we use for accurate sizing of objects, identification of objects, and with that came the advent of a whole need for software data integration, but also accurate geolocation to target what. So actually, the features we've seen with this SWOT mapper, we can actually target directly with optical sensors. And this brings on to the need to have these two types of technology, because habitats and ecosystems, they exist at multiple spatial and temporal scales. And you need to be able to zoom in quickly from one to the other. And so when you're first looking at biogeographic provinces, you needed the large scale purview of what sort of different sort of seascapes are out there. Then you needed the fine scale to say, well, what actually habitats were there, how vulnerable were they, what sort of spatial management measures do we need, and how can we implement monitoring programs. So the acoustics and optics were complementary methodologies to sort of get into an understanding of the seascape. I'll walk you through sort of one region, one study site. It's in, we call it the Southern Seamount study site. It's sort of experienced very large demersal trawling activity probably from 1987 to 2006. It was the area of the first seamount reserve for Deepwater corals in 1996, and that was extended in 2007 to a much larger area. It's on this, for those geographically challenged, that's Tasmania, which is in the southern hemisphere, and it's the bottom of Australia. So with the advent of the new sonars, we can actually map out these regions quite precisely, and we can start to see all the different sort of seabed features. And these are all these little bumps you see are sort of extinct volcanoes, and they all have very hard substrates. Prior to these mappings, we only had fishers maps of where things were, and some of our own maps. But we had no idea of the complexity and the sort of the number of sort of features that were in that area. You can see where the old MPA was. That was the first sort of seamount reserve. A lot of the demersal fishing was contained in this particular region here. And you can see that now the new MPA has actually taken quite a lot of that in, as sort of a no-take zone and no fishing zone. So our study that started in about 2005 was to look at the impacts of trawling as one aspect of it, but also to understand what is the biodiversity and what sort of communities are in that zone. So having sort of acoustic swath maps of that area is almost fundamental to plan out what your survey might look like. We also had, looking at trawling and understanding how the trawlers behaved in that zone at the time of the fishing industry, we also did a little bit of a synthesis of the trawling activities. And you can see this is some of the notes that were taken at the time about how they trawled, you know, what species they were getting, and it really sort of alluded that there are distinct trawl corridors that they had on these seamounts. Areas where they could fish and areas where they couldn't fish. Areas where they lost a lot of equipment and gear. Nets totally, sometimes people lost winches, which fathoms what actually happened to the rest of the gear. And so you'll see sometimes on our maps we have this little winch put in the map saying we think there's a winch down here somewhere, and with it attached to a really long cable. And when you're operating equipment, you've got to be, you don't want to get caught up in someone else's floats and gear, so areas would be avoided. But, it was quite a pioneering day, and some of the names of the beaches sort of reflect that. We've had to modify some names for public consumption, but most of the names we kept, you know, as the fishers had them. But the take-home message for us from this was that these fishers couldn't target these seamounts everywhere. So it wasn't a mining operation for corals. It was actually an economic exercise in extracting fish and getting benefit. So, so initially we had to sort of look at our sampling design to sort of make sure we took that into account. So we had a sampling design that sort of looked at areas that were heavily fished and other areas that were lightly fished, and then areas that were not fished. So the red's very heavily fished and still open for trawling. The blue is lightly fished. That can be a coined term. And the green is actually where there was no fishing. And you can see the video transects that were taken along that time, and this was done in 1100 to 1400 metres depth. And some of the publications are out of that sort of work. And what was found was that there was clear evidence for extensive removal of corals, and that's what we expected to see, and didn't know it occurred. It was, what we were trying to look at was the areas that were fished, and we also had a time series that we also had some areas that were sort of sampled 10 years previously. We're looking to sort of get ideas of recovery rate on areas that were lightly fished and heavily fished. So there's clear areas of total removals, and that was in an area that was very smooth bottom. We knew there was coral there from the fish's information, and that was completely converted as one would expect. In areas that were lightly fished, there were still areas that were very direct evidence of broken corals, stems sort of visible, and trawl damage. In the unfished areas, as you can see, it's quite vibrant sort of coral life, and so it was quite an obvious result. But what we did see was quite large areas, refuge areas where there had been a lot of fishing been occurring. There was still quite sort of vibrant coral communities existing. So I guess our observations have helped shape the paradigms that we've been hearing and seeing evolve in this debate. And there's the fished and unfished terrains were distinctly different, and so there's obviously a direct correlation between some of the fished areas and unfished areas and abundance of taxa. Our time series data was quite clear, that showed no evidence of recolonisation by the habitat. And that was after 10 years, and postulations are that some of the communities, especially the corals, would be many hundreds of years old, so there'd be a very long time to recover. So those paradigms were existing. the paradigm that wasn't interesting so much was that even in these heavily fished areas, there are large pockets of refuges that weren't sort of targeted by trawling, and that do have quite a lot of coral communities associated with them. And some species were sort of impervious, obviously flexible enough to not be affected by the trawling. So it was a slightly different paradigm shift from what we had expected to be 100% removed in these areas. And I guess from the Commonwealth perspective, they put this marine reserve in place, and so it seemed like a balance. They're striking a balance between preserving some of the corals, and though some of the acres was already been completely denuded, so why would you preserve it? But there are significant amounts of intact corals in that region, and it's sort of like that balance between utilisation and conservation. But there's sort of ongoing need to sort of reinvestigate these observations about exactly how much habitat is remaining in some of these areas. To give you an example of that, the green dots you see on the map is actually the new features that were discovered from our survey. So we had knowledge of all the other dots, the cones that had either red or orange. They were known about, they were sort of locations that were fished. The green dots were all features that we hadn't known about previous to the survey. So it sort of gives you an idea of the number of features. They're small, but they all have sort of hard substrate associated to them and potential sort of coral habitats and sort of biodiversity sort of communities or communities around them that have high biodiversity. So that was sort of an outcome, or sort of an unexpected outcome from the work. From the acoustics, we can get a lot of covariant data, such as slopes of these features, and in particular this backscatter, which gives you an idea of hardness. And hardness is something that the fauna uses to attach itself to the substrate. So having a knowledge of hardness gives you an idea of where the communities might be distributed along with depth and a few other covariates such as slope and oxygen content. And we came up with a system to look at the potential habitat range, or hard substrate versus soft substrate, and we've tested that with different types of biological communities, and it's holding it quite nicely. So the take-home message is that there's more habitat out there, and really we've only been sampling in areas that have been previously sort of known about. So the fishery questions and some of the things that we see as ongoing is, what is the actual habitat and what proportion has been removed? That's sort of an unknown to us at the moment. And what's been left intact? What removal or trawl use is acceptable? There's often a limit for 20%, so much sort of other strategy for the fisheries, but there's not so much said about how much habitat do you need to be intact, and how much can you accept to be degraded or sort of modified by your utilisation. There's a lot of new national and high seas regulations about benthic interactions and move-on rules and the like, and it's not so clear exactly how effective they are and what their outcomes will be. And we think the use of acoustic and optical technologies, both by ourselves but also by the fishing industry, will enable them to sort of bring data to bear to help address those questions. So that was the first sort of theme or chapter, which was understanding deepwater habitats. And the next chapter I'll move on to now is the fisheries associated with those habitats. And one of the dominant ones has been orange roughy, which has had a very colourful past, and it may have a colourful future, but certainly have a colourful past. And it certainly has been put up as a poster child of complete over-exploitation of the species. What can be done to a species in a very short period of time. So I'll discuss some of the findings that we have and some of the interactions that we see with the habitat. So that's the orange roughy there behind, for those who are colourblind. And some of the other species that are associated with them are not. The next theme I'll talk about is the food web that goes towards them. So some of the generic orange roughy population parameters. They're a deep sea fish. They're very long-lived. We've had genetic sort of aging samples up to 150 years. They have a late age maturity, and that sort of starts to bring a lot of alarm bells straight off about when there's being a sort of a harvestable stock. They have low fecundity, very large egg sizes though, and they form very large spawning egg nations. They're a batch spawner. And everywhere you work on them, they have uncertain stock structure. I'm not quite sure what the stock structure is. And just to put you in mind where we are, we talk about is the spawning populations around the east coast of Australia. This is the largest known spawning aggregation. And that's known on the east coast of Tasmania, bottom of Australia. And I've just been talking about some feeding aggregations of the habitat, which is in that southern seamount area of the bottom of Australia. There's a lot of different management zones, and of course the management zones aren't quite associated to the stock structure, which causes other problems in the assessment. And the take-home message, I won't elaborate this too far, but if you look at that sort of column, you can get an idea of where the species was as a function of B0. And under our new harvest strategy that was implemented in 2005, below 20% you stopped fishing. And you can see that, probably what's happened in most of these fisheries, it was well below in many of those categories. And as a function of that, the federal government assessed them and said they put them under a conservation dependent banner. So in 2006 they were declared conservation dependent in our waters because they've gone through a significant and adverse impact on their populations. And as part of that we set up one of the concerns of the government at the time was that they shouldn't go any further, and they did. They went through a sort of increased listing. And every time you go to increased listing, you have more and more management controls. So they were listed as conservation dependent, and if they weren't seen to be recovering, they would go to a vulnerable, and then endangered, and then critically endangered. And this would mean progressive closures of the whole fishery and any sort of activity in the Deepwater. And so as a result of that listing, we needed a rebuilding program to be put in place and a monitoring program. And of course, a monitoring program to detect something that's been rebuilding, when we can't even detect what status they were at, it was quite difficult. And a lot of times, in a lot of the zones, a lot of the information coming in was actually from fishers' dependent data of the catching and effort. And of course now, there wasn't that sort of information coming through. And we needed to show the stock was above 20% B0 or eventually reaching that level with 80% certainty. And this is where the acoustic slides sort of eventually come in. Acoustics was being used, and I've been using it since about 1989. I started off with these grey level recorders on fishing boats, trying to map out where these fishes were. There was minimal GPS coverage, and through my career I've been sort of evolving the use of acoustics, and by 2006 we had these new tow bodies with multiple frequencies. We were using them for species identification. We were towing them to deeply so we reduced dead zone problems and uncertainties of sort of sound propagation. So the science had advanced considerably, but at a great cost. All done by research vessels at that stage. And now there was particularly no money and no infrastructure to do that, and it was very expensive to maintain it. So even though we developed this technology, and I'll just give you an example here of how it worked. There's multiple frequencies shown here, 12 kilohertz, 38 kilohertz, 120 kilohertz, and a mixture of the three. And each frequency had a unique reflectance for the dominance of scatterers at the time. Here's these whiptails, which have gas bladders, where the 12 kilohertz was very strong for, and other gas bladders, for large gas bladders. The microneedle, which is a small sort of 9 centimetre, 5 to 9 centimetre fish. The 38 kilohertz, which is our survey frequency, happened to be particularly good at finding these things. You can see the orange roughly here. The 12 didn't see them at all. The 120 saw the orange roughly very nicely, but didn't see the tokens. And so mixing them together, you were able to discern the orange roughly from the tokens and then a large gas bladder species. So it was a nice way of sort of starting to segment these species groups. But again, we had developed this from research platforms. We had a lot of winches and equipment. So a natural progression in this stage is to move our transfer our methodology and technology to the fishing fleet. And I guess appropriately at that time, the governance and the motivation for industry engaging changed quite significantly. And we were getting very good relationships with the fishing industry, and the motivations and the sort of the incentives were being put in place to facilitate that. Of course, also working with the fishing industry, they had the capability to handle equipment, and the cost effectiveness was very high working with them. But still operating vessel amount of sounders from in such deep fisheries, it's not possible to do these techniques. So what we developed is another technology where we housed all our sort of multi-frequency methodology, our optics and videos, and actually house it into a unit where we put on the headline of the net. So we use their basic infrastructure and their knowledge of handling gear, flying it around these seamounts, and put our towed systems and put it into sort of the headlines of nets. We have updated this. On this slide, there's a whole lot of stereo photography and 2 extra frequencies. And this is the latest from this work, which is looking at the recovery in 2010. And you can see this is from their vessel-mounted sounder. You can see, for those who are acoustics types, this red pipe's the bottom. This echo here, this is developed from ships standing across the seamount. And you can see these sort of scattering layers here, which are the areas which is orange roughy, but a lot of other sort of areas. This is the AOS, Acoustic optical system, which is flown 500 metres deep. Just following the vessel, you can see these same features, and how they sort of are far clearer. We don't have problems with interference with the seabed as much. And you can see the sort of total layer here, and the sort of gas layer here. The 120 kilohertz is from the same AOS, and you can see how the structures are so much, the orange roughy structures are so much easier to see, and that's borne out when you mix them all together. So from this, we've removed one major part of the uncertainty in mapping these seamounts for species, which is the species composition. The next major uncertainty with acoustic surveys, and that's target strength. Target strength is the reflection of an individual fish, and to convert these acoustic surveys into biomass, you need to know target strength. And so this is very problematic for orange roughy. Often species have a gas bladder in here. Orange roughy has a wax ester bladder, very low reflectance. And you can see this is a density scan done in a hospital, Royal Hobart Hospital, at no cost. They didn't have a Medicare card. So quite happy about that. And you can see it gives a density contrast. And acoustics is all about sort of sound speed contrast and density contrasts. And from here you can see the density contrasts quite easily. You can see most of the density contrasts are actually in the head. And this is borne out from some simulations now by some very high resolution modelling. And you can see how complex the scattering regime is in the modelling. So to rely on median target strength from just modelling alone, there's far too many uncertainties about orientation and behaviour and the like. So what we developed, and one of the motivations for developing our AOS, am I supposed to be at a better lecture? We actually put the AOS, the ability to image individual fish, trawl down and actually, here's an image of, imagine that the net's going down, it's capturing fish. These fish are actually passing through the headline, and this is shown by the acoustics. The optics is showing the fish, and we've synchronised the optics and the acoustics so that we can identify a single fish, know that we've got an orange roughy, and one of the biggest hang-ups with doing this work before is that we could never identify positively what we were insonifying was orange roughy. So visual verification was very important to us, sort of like a quantum leap in sort of moving this science forward. And this is work we did in New Zealand. For anyone who knows politics of target strength in Australia and New Zealand, twain should never meet, but they did, and it's nice to see that we're both coming up with some very similar methodology and results. It also gives a bit more confidence to people using the data. We've now taken in 2010 that one step further. This is a very complicated slide. We have stereo optics on the headline of the net. We image individual fish. From that we can get their length, we can get their orientation, and almost sex stage and age, but that might be years to come. At the moment this is strictly just length and orientation. And from that we can build up data sets, and at multiple frequencies we're able to sort of describe the target strength as a function of a mixture of lengths and orientations at multiple frequencies. And we can start to develop a relationship between multiple frequencies and the frequency difference. And I won't go through the details. You have to trust me, being an acoustician and Australian, but it's true. But we meld that to our identification of schools. And our identification of schools is we use the frequency difference, and now we meld the way we do target strength and the way we do frequency difference in schools, and it constrains possible outcomes for target strength. So here's a way of locking in both our identification of the species by acoustics and their individual target strengths, and locks us into a curve that is quite narrow. We sort of physically can't move out of it. It's enabled us to come up with some very precise estimates of target strength that are matched to the schooling structures. And from that, now we get these new acoustic surveys, and it helps us to sort of put them into context of the assessment model that was happening previously. So now we're seeing a recovery and a status of orography. It's not hugely different, but it's subtly different enough to say, well, both the model is predicting an increase, and we're observing an increase on these spawning seamounts. It's quite clear. The difference is that the acoustics is higher, and that's just the snapshot spawned population. It doesn't include portions that aren't spawning and turnover rate. So it holds some great promise that this fishery is recovering. But obviously, people don't actually always believe in acousticians when they talk about biomass. And I really don't understand why, because we're always so good at it. But in the past, there have been some major mistakes, and so obviously you'd want some other confounding information to be helpful here, but it's very good evidence to say that this ground, that the fishery is recovering, and we perceive to be a lot higher biomass than expected. Now, there's been a lot of literature being put through about orography and its ability to recover, and I'll just go through a few indices of other things apart from acoustics about how they could recover. This is a plot of age of orange. This is the mean age of orography at the start, pre-fishery, 1987. Mean age about 50 to 55, 60. And I think probably the important bit is just to look at 2010, the mean age is around 35. Now, this fishery is based on the spawning population, and the only way to get an age shift, really, unless you're very selective in your fishing capabilities, is for recruitment. But you can also see major differences between these two sites, the spawning sites, very close to each other. And the noise is associated with just the age data. And we can see at sites that there's age segregation or size segregation between sites. An index that commonly gets used is length. And this is the same sites. St Helens is one, St Patrick's is the other. And you can see the length, there's very little change, about a centimetre over that time frame. But again, there's quite a lot of variations and differences between sites, which could be sampling bias or could be real sort of age or length differences. An unexpected finding that we had is that we did we repeated a study on the eggs per gram of ovarian tissue. We repeated something that was done, luckily for orography, which is probably a rare species. A lot of work on orography was done prior to the fishery being totally developed. 89, 90 when the fishery took off. So we have a lot of data in this 87 period, 89 period, which is pre-fishery. So we had this idea of standardised fecundity going through, and one of the tests we wanted to do was just repeat that study to see how it was evolving over time. And we came up with this number here, which was quite, it was very surprising. And this is basically, there were more eggs in the yolk, so actually there was less atresia happening. And the condition of the fish were far much better. And so there was this thought, at the time, a hypothesis was put forward in 92 of density dependency. And it's one of the sort of hypotheses that still occurred as the fishery got decreased. And we think, whilst this is nice to see here, obviously you'd like to repeat it. You can see that it was done in 92 and 2010. So you can draw in it's a lovely line. But we're suggesting that this should be done more regularly over the next few years to sort of prove this up. It seemed a very positive result. So for our status of orange roughy, many industries are showing a stock size of biology that's showing a marked response to fishing. There's evidence that there is recruitment inferred from this age data. There's evidence of density dependency. And the acoustic surveys are showing that the biomass is stronger and there's potential recovery. We think that it's been good for the conservation dependency. I don't think they'll be put into the next level, and we're recommending that they sort of continue a monitoring strategy over the next few years for this species. Now, I'd like to contrast this with a study that was done in New Zealand, which is after 20 years of fishing, where they found no change in the GSI, the gonad index, whereas, funny enough, we didn't see much either. It was actually the atresia or the viable eggs that had changed. There was minimal change in length they observed, and again, we haven't seen change in length, but we've seen a major change in the age structure. And there's a lot of debate about aging error, and I could go into that now. These ages have been re-aged and to ensure consistency over time, which is one good thing about having an open database. And they inferred from that there was maybe due to poor recruitment and there'll be a long time to recover. And it's something that we haven't, we're finding that that's not the case in our waters. So another paradigm might be that not all stocks are the same, and not all stocks are behaving the same. So that was the second sort of theme of my talk, was the orange roughy fishery and some of the sort of the latest data on how this stock is potentially recovering from fishing. I'm going to move now to the next chapter, which is the mesopelagics. So around these seamounts is a mesopelagic community, and what we call the micronekton. And these are very expansive. They cover the world's oceans. I'd like to sort of describe some of the sort of observing system we have for that. Right. And part of the motivation, and I guess I put this slide up because it is motivational to me, is that people are developing ecosystem models, and they're trying to predict primary production and secondary production, and sort of epipelagic and secondary productions through the world's oceans. And they're doing that sort of with obviously coming up from physical forcing fields, coming up with primary production from satellites, sensing, and then they're going into estimating epipelagics and using some basic sort of growth and growth curves due to temperature here from historic data on sort of growth rates. And I find this sort of inspiring because this is sort of getting to understand sort of the global ocean and understanding what sort of other trophic groups you have past primary production and eventually get to top predators. So there's quite a few of these. Patrick Lahoyder and his team have been doing this particular one. But what they do need is initialisation and assimilation of data. And these models are probably about where the physical oceanography was about 20 years ago when they started up their observing programs. So in our waters, some of the background is this missing link. Often these models get developed, and what's missing is this mid-trophy. We get to the primary production and they get the top predators because they are well sort of monitored. But what was missing was the micronekton. And this was sort of seen as needed for these ecosystem fisheries management, marine planning, and for climate change. And also predicting shifts of communities in future sort of climate variability, climate change scenarios. So what we put forward is developing a basin scale sort of monitoring program around, especially in this case, initially started off between Australia and New Zealand, because that was seen to be a place where predictions of temperature rise were the highest because of extensions of the East Australian current. And we said we needed a biological baseline for that area. And one of the ways proposed to do that was through this sort of via acoustics from ships of opportunity. And using these deepwater echo sounds, you can see here, sounding in the ocean, coming up with these echograms that are representative of some of the biological life in that, in the open ocean. And these go down to 1,000 metres. And this is typically what the makeup of that is. You put a net down through some of the deep scattering layers. What you collect is a range of light fish, hatchet fish. I think there's a tubia here, some gelatinous, a lot of gelatinous material. These little fishes, you probably can't see them, but they're called gelaphants. That's their official name. So they've got this big snout and lie, and yeah, so that's a new species. I'm sure it's not, but that's what I call them. And these are crustaceans. So they're the sort of things that are in those deep scattering layers, and the things that sort of we're attempting to monitor. So when we started this program back in 2004, 2005, proposed to use them as a ships of opportunity and using fishing boats. And predominantly it was all fishing boats. Now we've started the program, we have research boats on board as well. Research boats are hard work. Fishing boats seem to be a lot easier. They don't argue as much about what to turn on and off. And these tend to be a rapid and cost-effective method of sampling. You can see from the data from this one, it gives you diurnal information. This is the organisms moving out from the daytime habitat, up into the night time to feed on the plankton surface, and of course going down again during the day. And that's a diurnal cycle that happens right through the world's oceans. There's a depth sort of distribution of these biota, which can be described by this, the acoustics. Geographic was unfortunately New Zealanders that they had low backscatter of mesopelagics, which aren't economic on their waters. We had quite a lot of them on our waters, which hasn't really upset them much, but it was quite intriguing. It was quite the opposite was thought, actually, that the density was uniform, but it's not. It's actually, the biomass is seen quite lower on the New Zealand sort of side of the Tasman and a lot higher on the Australian side. And there's interannual variability. And also localised. We have a lot of eddy sheeting on the east coast of Australia due to the East Australian current. And we get these warm water and cold water eddies, and they're sharp. Probably this is a very fine scale, but in sort of low, sort of the actual it shows quite devoid of life actually inside one of these eddies. And most of this is coming from the gas bladers of mitomas, so that's what we suspect. And the other species groups are the jellies, the squids, and the small crustaceans. And one of the tasks is not only to have this sort of remote sensing, but actually to try and quantify some of the biomass of those scattering layers. So this is an example of, we started this program with the Australian Integrated Marine Observing System in 2010. And this is some of the tracks we have from 6 vessels, 6 fishing vessels and 2 research vessels that are steaming around the ocean doing repeated transects. The other advantage of fishing vessels is they usually, their motivation is to repeat those transects every year to go to their fishing grounds. Whereas research vessels only repeat their transects based on what researchers want to do. So fishing vessels again have a bit of a plus, which is these sort of observing systems. The data is now available on this web portal. We had to sort of solve a lot of analysis problems, a lot of metadata problems, and developing standards. All things that are needed for an appropriate sort of observing system. And like I say, we have worked through a lot of those issues, and they're still evolving, but most of them are starting to sort of come to fruition. And you can click on that and you'll get the transect and actually an image too, to sort of see how the backscatter is evolved across the transect. Another thing that we've been doing in the observing system is actually co-linking other sort of biological methods. And here we've co-linked the plankton, sort of continuous plankton recorder, which we have on some of the selected transects we run. And we run them coincidentally with the acoustics. And one of the motivations of that is to then cross-link a range of data sources, so that we're not talking in 10, 20 years' time, I wish we had these types of data sources. They're available and they will exist. So truly it is setting up a decadal program of observations of the biofilm, not just the physical environment. So this is again the transit across from Australia to New Zealand. It takes 3 days to ship, 1, 2, so it's 4 days almost. You can tell that by the diurnal migration pattern. So there's 1 day, 2, 3, 4 days to get across the Tasman. And they were tying a plankton recorder, and as expected from both ocean colour and our own acoustics and and physical sampling that we've done, there's more biomass on the west side of the Tasman than the eastern side. Funny enough, the cable pod density was the opposite, so the trophic exchanges, trophic linkages are quite complicated, and, but at least we start to evolve the data set where you can start to sort of tease those things out. So I'll talk a bit about, it's really nice having these images and this sort of data to qualitatively look at. And one of the things is obviously it's going to need some data interpretation and uptake. And this is an evolving field, and whether you can use it as this data as straight as an ecosystem metric for distributions of communities, and under what conditions can you use this echo strength directly in proportion to some biological metrics such as calorific content. In some cases it's been shown that that's true, that you can convert acoustic content straight to a biological metric. But that's usually under some circumstances. We've started the integration with models, and already with Deepwater, we've started an exchange of data, trying to put into the models, trying to develop sort of data assimilation methods to inform what is the epipelagic, mesopelagic, bathypelagic, and the energetic exchanges that occur, both daily but also seasonally. And of course, the all-important is the extra validation research and the ongoing need that we've got integration of new technology. So for validation, I'll just quickly, if I may, go through a bit of validation, which is we have this open and closing protein net we put on the back of fishing vessels and research vessels, where we capture organisms through the water column. We also have probes that we lower down to get detailed acoustics of the organisms. And this is how it works. You put the net down, and then these nets open, and they fish through these layers, and you capture organisms in each of the nets. And then you can relate that back to the acoustics and any of the other sampling devices you might use. Now, everything's selective, and everything sort of has bias. The nets are biased due to the species, to the cod mesh sizes, that what gets retained, avoidance responses. The acoustics is also very selective because it only captures things that are reflective, and things that have large gas bladders get easily seen. Things that don't, like gelatinous material without gas bladders, don't get seen. So there's a lot of biases in all the observing techniques that we use, if not these due to avoidance. So with acoustics, we lower through the water column, and we get, this is the acoustic probe lowered through the water column. It has a standard sphere in our acoustic circles that's sort of hung beneath it, which is constantly calibrating the system through the range of depths, has no reflectance. And all these little squiggles here are individual targets, and they're individual sort of recordings of an animal that we recorded. Now, this is at one frequency, and now we do it at multiple frequencies to try and decide what species we're looking at. Now, the current status is that there's a huge difference in estimates of biomass. A lot of the models that are using are coming up, and I've just got this area here as an example, it's the Tasman Sea. They would estimate between 2.1 to 8.2 million tonnes of mesopelagic or total biomass. Nets, we did a net sampling study, you come up with 6.7 to 10.7, and acoustics at a whopping 6.4 to 11.19. So the state of the science is, I can't tell you right now which one is correct. Which is a bit sad, but I think that's just being honest and saying that's it. You know, we're just starting this road of understanding of what is actually up there. And we've got quite a bit of work we've done already. It's helping us to sort of define that a bit better. And this is the type of work, and I talked about it with the orange roughy. We're applying the same philosophy where we're using nets, we're using optical visual methods, and we're wanting to combine that with our multi-frequency to improve our species ID. We want these depth stratified netting, not just for the species, but also for the biodiversity. A lot of the length, weight, life history parameters of the organisms, their isotopes and genetics. So we've had a program involved that's been collecting that data and we're just working through it. And very importantly for acoustic is this visual verification. I think that's the key for us to sort of understand the reflectance of these organisms. An example of some of our netting going across the Tasman, you can see in different parts the changes in the length structure and the community changes. And there's quite high diversity for Deepwater anyway. We had 32 species of tophids in that year recorded, but only 2 or 4 species are making up 70% of the biomass. So there's quite a lot of dominance in different species, which sort of then makes it a bit more tractable from a remote sensing acoustic point of view and concentrating on the dominant species. The next is the images or biota. And a lot of things with the gelatinous material, we don't actually see very well with the nets. Anyone who's seen a gelatinous material come up in a net, it's extremely difficult to tell what they actually were. And anyone who's a non-specialist. But in the images, you know, I show these images to taxonomic people who are, and they just think they just say what they are just almost straight away. And so it's quite nice to get those optical images and then have them pointed out to what they look like in the tray. And so even a non-specialist can start to say, oh, that's what it looks like, but actually it's all smushed up and you can't sort of see much of it. But now some of these critters are siphonophores, and they have the most unfortunate habit of having a nadphore, which is a small gas inclusion, which reflects very highly in acoustic frequency terms. So if there's a lot of siphonophores in your region, it can quite fool you into thinking that there's a lot of gas-blooded species that actually produce gelatinous material. So that's why we tend to be a bit sensitive about gelatinous material as in an acoustic sense, apart from their ecological role. And of course we refine our methods, looking at gas bladders and coming to the different dominant species, trying to refine our multi-frequency methodology. I better just move along, I'm running behind time. And so this is all wrapped up in sort of a global context. And I started this with looking at it in terms of the Qualitative lineage, which is the climatic impacts on top predators. And there's a white paper out about how to look at this in a global context. And I've just been concentrating on the ships of opportunity. There's many people here that have been doing moorings, from Horn has, and also people looking at these other aspects of deploying acoustics on these platforms. So from the bioacoustics, the ships of opportunity, we've started this decadal program. It really is just the start. We've proven that it's a tractable problem. We've proven we can do it, and we're hoping when it comes up for renewal in 2 years' time, the whole program, that acoustics will be a major player for the next 5 to 10 years of this opportunity strategy. We've commenced this integration of the ecosystem models, and we've started our road onto this validation and visualisation of the data, both nationally and internationally. Right, that probably leads me to conclude, and probably I'll conclude in those 3 major themes I talked about. That's deep sea habitats. Now we know they've been heavily exploited in parts, but large regions are unexplored, and the impacts of human activity is not well understood. And that's both direct impacts from trawling, but also indirect impacts from ocean acidification. We have some recent findings of benthos at 2,400 metres, and it's just at the tip of the iceberg of us sampling Deepwater beyond fishable depths, which normally we wouldn't have gone much below 1,500 metres in our work. Shown that seamount habitat is slow growing with a long time to recover, and direct impacts such as commercial fishing. but the demersal fishing is targeted, and whilst destructive, it's not 100% extractive. And acoustic mapping and visual sampling is a definite need for our ongoing understanding of deepwater habitats. So this is a sort of an example of the latest work from Ron Fresh and our team, 2,400 metres, showing this extraordinary benthic community that just about to publish another one that shows the high diversity of these depths. Put that in perspective, this was taken from this little spot here. Our seamount study site was here. These are the MPAs that we declared in our water. This is our EZ net, so that's 200 nautical miles to here. To give you an idea, that's the terrain that's available. This is just one small spot for the study. And if you transform that sort of bathymetry map into a backscatter map, which starts to tell you something about the substrate, you can see the available hard substrate for attaching communities. Not talking about the burrowing communities, just the attaching communities. And the dark is the actual hard substrate available for attaching communities. And it sort of gives you a sense of how much we haven't sampled and how much so, consequently, potentially how much we don't know about our own region. But certainly having these maps can start to give us an idea of where we should start looking at sampling. So the next one was the Deepsea fisheries. I really have to move along. And that is the overexploitation of orange roughy. It demonstrates the effect that modern technology can have in decimating a species within a fishery within such a short period of time. I think most of the damage in the orange roughy fishery was done in 3 years. 3 years of unconstrained management, and the technology was just available. And the GPS had just come online, modern trawlers, modern electronics, colour echo sounders, and headline units where they could find out where their gear was. And in 3 years, you could use that power of 20 boats to almost decimate the fishery. So the management measures were slow, but the governance dramatically changed. I think one of the biggest advantages I've seen in my career is this improved governance structures that have been put in place. And they're now being implemented, especially in Australia, in my knowledge, the Australian region. And our acoustic and optical devices are now providing new insights that maybe it wasn't as dire as we thought. That whilst there was a lot of removals and a lot of habitat removed, there is habitat left, and the orange roughy might surprise us and actually recover quite well from this, what has been a very dramatic decline. And the last one is these, what I call the frontier for my work, is this basin scale monitoring, and that's these models of ecosystem which are just inspiring and sort of motivational and sort of getting out there and getting an observational program to sort of understand and test these hypotheses that these models are coming up with. But the need for these well-funded sort of scientific monitoring programs to validate and sort of initialise these models. And how our understanding of this basin scale is going to be very, I think, very informative for us in a changing climate. And should be a sort of area of active research in the coming years. And the humble acoustics and optical methods will play a vital role in that area. So thanks very much for listening. I'd also like to thank the Bevan series for inviting me here, and especially Trevor Branch, John Horn and Sri Rangan for making me very welcome. And also my colleagues from the Deepwater Ecosystem Status Prediction team, our IMOS Acoustic team and the Oral Practice system. Thank you very much.
There's probably time for maybe 1 or 2, maybe 3 questions. I'll hand over to you and you can select the lucky few.
All right.
Yeah.
Okay. So I hadn't meant to go over time. Sort of evolved.
Well, I have a question, which is, it's often been said that there are 2 remaining big, untapped resources of fish in the oceans. And one of them is Recolonisation, the other one being Antarctic prawn, which is the station.
Yeah.
But nobody really seems to know how many Recolonisation there are. It seems like your work suggests there could be vast numbers.
I think they're sparsely populated. They extend over such a wide area that it doesn't take much per metre squared or per kilometre squared to suddenly add up to a really large biomass. But I hadn't thought about it in exploitation terms, I guess. But I guess someone will.
They're very unpopular.
Yes. Chris?
Yeah, I thought that, I want to ask you a question about the first of the three.
Yeah.
You mentioned that the recolonisation by Deepwater stony corals is basically not happening at too great a rate. Did you have the opportunity with your acoustics and other monitoring devices have an opportunity to look at whether those heavily trawled areas were being recolonized by other species of fish in a relatively rapid rate? I'm wondering if you changed the bottom habitat, you improved the stony corals. And I'm just wondering if there's now other species of fishes that may be moving in and recolonizing those heavily trawled areas that are no longer fished heavily, or not.
I think given the studies on it, it'd be hard for us to make any sort of comment about that. And in terms of the initial study was just by drop cameras, and not necessarily, you didn't have the towed videos where we actually do have fish counts, and we actually have populations of organisms actually on there. As far as colonisation was, there's a picture I showed of the sort of soft corals that seem to be very pervasive in these regions, and we couldn't decide whether they were new colonisers or they'd always been there and just not taken off by the trawlers. And I think the safety wisdom from the taxonomists were that they were always there, but I think there was still some sort of uncertainty about whether they're just those stalked, sort of soft corals were actually new colonisers. So to answer the question, no, I don't know if we've got enough information or can say anything informed about the species recolonisation, or whether, even after we moved all the orange roughy, whether other things are moving in. And one thing we do see is a lot of deepwater sharks. They're pervasive in the depth ranges that we work, and they seem to be very, very numerous. And I wonder, one of the sharks we have do predate on orange roughy, and so there has to be some ecological consequence, right, from removing that amount of fish. Something else might come into that space, but we have enough information to say.
So given the range of years that have been used looking at orange roughy biomass on Deepwater fish, given the assumptions and constraints of all the years, how do you resolve, or put another way, who's got it right for ecosystem models, direct sampling, and remote sensing? Because that's not necessarily unique to orange roughy, and a common constraint to any sampling here.
And I think it's a challenge about how you integrate sort of, or sort of look at different sort of conflicts in data and how that gets handled. And I guess that's one of the things I've noticed where I'm being engaged at the moment is how do you look at these conflicts of data? And clearly that's what's happening in the model. It's not the model saying something, it's actually the data inside the model that's giving a conflict of outcomes. And it's also about assumptions of stock structure and a range of things. And I guess my belief is that science should come forward with the evidence to demonstrate that the techniques are robust. And I think that the orange roughy story we will put forward, and I will put forward, I guess, that we've locked off a lot of the uncertainties and we're sort of narrowing down the uncertainties. Someone else might look at my data and say, oh, they're over-optimistic, there could be bias there. But I think in a burden of proof context, I mean, there should be some sort of, if people can repeat studies in various regions, and what we've done in New Zealand now, or the colleagues in New Zealand, if they repeat the same work and come up with similar answers, it sort of adds weight. Okay, maybe you've got something here. It was similar to how the age of orange roughy got established. There was aging studies of orange roughy in the early years, either 10 years or 100 years. So quite a lot of work went in place, and eventually it's coalesced into a knowledge of age that everyone sort of understands, and it's been validated, I guess, and it's quite well understood. There's not many arguments anymore about aging of orange roughy. So I don't know if I've answered your question directly about how you deal with aging conflicts, how you deal with conflicts in data. But the management outcomes are quite clear. They'll go to a precautionary approach, as they should. And so the burden of proof is definitely on the more optimistic outcome, as it should be. And so that's why we're saying you should repeat it. You should sort of, before you move from one sort of precautionary sort of assessment, you should then sort of have quite a lot of justification for cover. We don't want to go to the sins of the past where everything's quite optimistic about a lot of outcomes. Orange roughy and a lot of other species. Sorry, Jeff, and then
are your ships an opportunity to use as government charters, or are they used by the industry to support the resource?
So the motivation of the ships and the industry, yes. I guess that's the question. What the government structure has brought about, I think, in 2005 was a shift of the burden of proof from government scientists, government sort of organisations to industry with the precautionary approach. And with that, a change in behaviour of the fishing industries and how they behave, the assessment meetings and monitoring. And what happens now is that because the book, in this example, it's a clear case, the precautionary approach has been applied, there'll be a low quota or a conservation dependency put on them. And it's really up to the industry to come up with a monitoring program and platforms to prove that the fishery is recovering and that it will recover if not recover.
Now, do you have government observers on there? Or?
Yes, so they, while the system is facilitating that, they still have to, in this case what's happened is there's been a research allowance or a catch that's been associated to this operation. And so the industry have to sort of work with scientists to develop a monitoring plan, an observing plan, and then implement that and get it validated and approved at sort of the fisheries management level. So there has to be a research proposal put into place, plan, and they get allocated so much research quota that they can use. And so this is not being funded by management or the management system, but it has been funded by a research allowance.
And the data is then processed by the science?
So yes, so I guess the scientists are still in charge of the whole project plan and the operations and the implementation of it, yes.
One last question?
So, yep.
The species that we study don't typically care so much about international boundaries like we do. Are the bodies responsible for governing areas where these species are harvested? Do they tend to play nice and see eye to eye on what the appropriate conservation management strategies are? Or is there...
Right, welcome to the world of RFMOs, Regional Fisheries Management Organisations. Having been in the South Pacific Regional Fishing Management Organisation meetings, we sit amongst a range of countries. In this example, it was from Chile, USSR, China, Japan, Korea, Australia, New Zealand, a range of players. I can say without any hesitation that there's never a consistent message that comes through. But I think these regional fisheries management organisations are coming to fore, and they are making great inroads in people playing nicely and doing the right thing. I don't know if they've got enough teeth to do it now, but certainly from my experience in being involved in it, setting up high seas marine protected areas, I think is a real challenge, but I think it's going to come about. Setting up some behaviours of how international fishers operate in high seas and move on rules and governance for individual states to abide by. I think it's happening. Probably very slowly, but it's happening. I don't know if that answers your question.
Well, we had some drinks and light snacks.